So this is our first session where we are going to do a comprehensive review of pretty much everything on the MCAT. And so what we're starting with is content category 1A of everything on the MCAT. So 1A is structure and function of proteins and their constituent amino acids. And this is a huge topic, like throughout everything. You'll see this pop up in every section. So this is something you really want to know and you want to know it well. So today what we're going to do is do a brief overview of all of this stuff, all of the important points you need to know, and then we'll do some practice problems and see if you actually, picked up on it and understood it. Okay. So I found this site that I think is really, really nice for just a brief explanation of like the high yield stuff that you need to know for amino acids and stuff. So we're going to just dive into it. So first things first, and also, yes, if you haven't taken biochemistry, some of these things might be new because amino acids, I think I learned for the first time in depth in biochemistry, but these are going to come up and you need to know them really, really well. So let's start. So first of all, amino acids are the building blocks of all proteins. So proteins, as we know, are polymers. They're made up of many units. But amino acids are monomers. They are the simplest form of a protein. And amino acids have such great variety and they inform the properties of the proteins that we have. So they're really, really useful to know. So the structure of an amino acid looks like this. There's an amino group, there's a carboxylic acid group, and then there's two groups here, a hydrogen and an R group. Now the R means that it can be any kind of group. There's a variety of different groups, and whatever the R group is informing what kind of amino acid it is, what kind of properties it is. The unique identifying side chain is the R group. Any questions so far? And if you have a question, usually we just drop it in the chat and then I get to it. So if you have a question at any time, just put it in the chat and I'll see. So an amino group is this group right here, the NH3. The carboxyl group is the COO, and the H is just the H, right? And protonation. Okay, we'll talk about that in a bit. Okay, yeah, this is important. Zwitter ions are things that have a positive and a negative charge, but in such a way that they cancel out. So at physiological pH, right, this N has a positive charge and this O has a negative charge, right? Just because this N is not, normally it's supposed to be like NH2. If it's an NH3, then it's an NH3 plus. And this O has 1, 2, 3, 4, 5, 6, 7 valence electrons. So it's a negative. But because there's a plus here and a minus here, the overall molecule, the overall amino acid is neutral. And that's what we call a Zwitter ion. And this gives us a lot of implications for how they can behave in our bodies. So now moving on to the complex part, the R groups. The R groups are what determine the identity of the amino acid. And the central carbon is called the alpha carbon. These are so this is this right here, the one that all four things are connected to is the alpha carbon. So chirality I'll talk about in just a bit. So here we can see the part in blue is the R group. So notice here we have the carboxylic acid, the amine group, the H, and then the R group. And the R group changes. It can be a hydrogen, it could be a methyl, it could be an isopropyl, it can be, I don't know what this is called, but this is 4 carbons, and then this is 1, 2, 3, 4 carbons, but in a different configuration. And so the thing is that you need to know amino acids really, really well. You need to know the structure, but the structure is basically this pattern is conserved and it's just the R group that changes. So you need to know the R groups basically. Then you need to know the name, the 3-letter abbreviation, and the 1-letter abbreviation. So this seems like a lot, but there's a lot of like mnemonics and stuff that you can use to learn it and memorize it. Not easily, but it is something that you're going to use so often that you're going to get to know it really well. And the other important factor is the properties of the amino acids, because in biology it's a very common thing. My professor always used to say structure leads to function, right? So the structure, the actual compounds, are what informs the function of the amino acid. So these guys, CH3, right, these are non-polar side chains. They're non-polar. So that gives you an idea of like where in the body we might find, or where in the cell we might find these non-polar amino acids that don't have any, negative or positive charges. They are just, and in this case, this one's aromatic. So that's another property. So these guys are, and this one has an OH, but because of this giant aromatic group, this is overall considered nonpolar. So this is the nonpolar group of amino acids. Then there's the polar group of amino acids. These guys have polarity, right? And can even be, these guys, these serine, threonine, and which one is it? Serine, threonine, and tyrosine, right? These guys, because they have OH groups, they can actually be phosphorylated. And that's something that comes up as well. Let's see, phosphorylated serine. This is not what I want. I want the amino acid. Here. So these guys, notice how this is the carboxyl group, this is the amino group, and this is the H here is invisible, and this is where normally it would be an OH, but instead there's a phosphate group attached to it. So this is a phosphoserine. So that is a question that comes up. Which of these amino acids can be phosphorylated? And basically, if it has an OH, if it has a hydroxyl group sticking out of it, like this, then it can have a phosphate group attached to it. That's just something like that is going to come up at some point because of how important phosphorylation is to activation and inactivation. So these are polar amino acids. Polarity just referring to the fact that they have like an electron. Does that include threonine? Yes, threonine has an OH. So this can be yeah, threonine is right here phosphorylated. So threonine, serine, threonine, and tyrosine are the three amino acids that can be phosphorylated because they have this free hydroxyl group that can attack a phosphate. So, and then we have these guys, cysteine, asparagine, and glutamine. Now here's where it gets a little weird because you'll notice that the G here, the glutamine, the one-letter abbreviation is a Q instead of a G. Why is that? Because the G has already been taken by glycine. So it can't be G again. So unfortunately, I don't know why they didn't call it G2 or something, I guess, but unfortunately it's Q. So I often think of this as glutamine. But whatever way that I'm just pointing out that it's not super intuitive sometimes to remember the one-letter abbreviations, but the one-letter abbreviations are important. They do ask about the one-letter abbreviations. And so now we have acidic side chains. So what does it mean it's an acidic side chain? Well, you'll notice that this is an O minus. Why is it an O minus? Because it was originally an OH, but it gave up the H. And what's an acid? An acid is something that donates its proton, right? So this is an acidic side chain because it gave up its H. So it's negative. And this is these are the two acidic side chains, and these are the pKa's. The pKa's become relevant when they ask you about protonation and deprotonation. And basically you'd be thinking like, will this be protonated or deprotonated at this pH, right? So if this guy has a pKa of like about 4, right? And it's in a solution, it's in a buffer that is pH 6, right? Is it going to be protonated or deprotonated? Well, the easy way that I like to think about it is simply by thinking of it in terms of numbers, right? So let's say we have a pKa of 4 and we have a pH of 6. Which of these is the bigger number, the 6 or the 4? The 6, right? If the 6 is a bigger number, then I think the 6 is a bigger one, so it gets the proton. So if the 6 gets a proton, that means that this is deprotonated at a pH of 6. Now, if it was at a pH of 2, right, and we can even think about it, pH of 2 is highly acidic, right? 2 is smaller than 2 is smaller than this 4. The 4 is the bigger number, therefore the 4 gets the proton. Therefore, this guy would be protonated. Does that make sense? What I just talked about with the pH and the pKa? Okay, so if we're talking, so this guy, so pKa is a measure of, how likely is something to give up its proton, right? So if this is originally an OH, right? At a pKa of 4 and above, he will give up his proton. At a pH of 4 and above, he'll give up his proton. At a pH of 4 and below, he will hang on to his proton. And the easy way to remember that is just by comparing what is the bigger number. If the solution, so we're talking about this amino acid, this molecule, versus the solution that it's in, versus like the solution of buffer that it's in, right? I'm just gonna poorly draw some buffer here. So this guy, this this oxygen here, this OH here, has a pKa of roughly 4, and the buffer has a pH of roughly 6. Since the 6 is bigger than the 4, it wins the proton. That's how I think of it. That's just an easy way to remember it. It will take the proton away because it's the bigger number. That's how I think of it. What's the actual explanation is like much more complicated, and we can get to that when we do, pH and stuff in chemistry. But for now, it's good enough to know when will something be protonated or deprotonated. And it's going to be deprotonated if it loses this like number battle, right? But if it wins, if this is actually like a 2 or something, 2 is highly acidic on the pH scale. On the pH scale, right? Like it goes, if it's 14, it's very basic. And if it's like 2 or 1, it's highly acidic, right? So if this is a highly acidic solution, then this is the bigger number and this gets the H. So at a buffer of 2, this would be protonated and it would be a hydroxyl. Does that make a little more sense? Okay, cool. So any questions before we start talking about, well, any more questions, I guess? All right, so now, and then we have these basic side chains. So basics are just the opposites of the acidic side chains, right? These guys gave up their protons because they're acidic, but these guys hang on to their protons because they're basic. They accept a proton. Rather than hang on, it should be they accept a proton from the solution, right? And it's the same thing with the pKa here. These guys have very high pKa's. So if the solution is a low pKa, they're very happy to take it. They will only give up this proton if the solution of the buffer is a very high pH, a very basic pH. And so basically, it's important. There's nothing here that's not important to know. Amino acids are probably the most high yield topic, and that's why the AMC is just giving it to you as like the very first foundational concept. There's nothing here that I can say is like, oh yeah, you could get by with like not knowing this. Everything here is like fundamental, absolutely fundamental. I'm talking about drawing the amino acids. I'm talking about knowing the names. I'm talking about the pKa's. pKa's show up a little bit less, but it's still really important to know. So and especially the properties, like what is a basic thing, what is a acidic thing, et cetera, et cetera. Any questions on this? And so basically here they talked about, nonpolar, polar, acidic, and basic. And they just talked a little bit more about it. Also, I'll give you this link in the chat if you want to look at it. It's pretty well written. Oh, before we go here, let's talk about chirality real quick. So chirality is it's from organic chemistry, so we won't talk about it too much, but basically if a molecule has this is a carbon, if it has 4 different substituents that it's attached to, that means that it's a chiral molecule. That means that it has different like isomeric configurations, stereochemistry, right? And basically all of them are chiral except for glycine, because glycine is the only one that does not have 4 different unique groups. It has 2 hydrogens, so it's not chiral. As well as this little fun fact, the chirality there are only L-handed amino acids, not D amino acids. And on the other hand, there are only D sugars and not L sugars in the human body. But that's something more relevant to organic chemistry when we get to it. Okay. So amino acids, peptide bonds. So now we start talking, remember, this is about proteins, right? So how do we get from amino acids to proteins, right? And that is through this nucleophilic substitution reaction. So basically, we have this right here is a typical, again, this is not an amino acid that we know of because there's an R group here, right? So this is just some amino acid. But regardless of the R group, the mechanism for the peptide bond formation is the same. And a peptide bond refers to the bond between 2 amino acids. So here, what happens is the electrons on this nitrogen reach out and attack this electrophilic carbon. This carbon is electrophilic because of this O group like sucking away the electrons from the carbon. So this attacks here, and then we have this situation where if you this is a little bit more into organic chemistry, but I'll just explain it because I like organic chemistry. So the electrons here attack here and push this. Oh, let me just draw it. I can draw it. Push these electrons up onto the O, right? And so now the O is negatively charged. And now we have this situation where it's like these 4 groups are attached, but this is not sustainable, right? This is way too stereochemically crowded. So what's going to happen is that the negative charge drops back down, and then one of these has to leave. It's not going to be the carbon leaving because carbon can't have a minus charge on it. It's not stable. It won't be, it could be this whole amino acid leaving again, but that's not what we're doing here, basically. The best leaving group here is this negative OH. So once this guy leaves, we have the situation on the left here, which is our newly formed amino acid and this free-floating OH. So let's draw out what just happened. This thing right here is our original amino acid. And this guy right here is the second amino acid that just joined in. And now we have a dipeptide. Now we have 2 amino acids and we have the start of a polymer, right? It's just a dimer actually. Right now it's just a dimer. Then if another one joins, it'll be a trimer, and then so on until we get a polymer, which is a polypeptide chain, aka a protein. So that is how, one at a time, they link up and become a protein. And so you'll notice the important thing is that a molecule of water is produced. This is an OH, but it'll pick up like an H. It'll pick up a protein from somewhere and it'll become a water, right? So a molecule of water was produced. This is a dehydration reaction, I believe. Yes, I think this is a dehydration reaction because water was produced. Now the opposite is a hydrolysis reaction where you could have this. If you did all of this in reverse, then you would break up the protein, right? So if this O minus decided to come back, everything would happen in reverse, and then we would just break up into two separate amino acids again. So that is the mechanism behind the building, and also, if you go in reverse, the breakdown of a polypeptide chain. Any questions about this? Okay. Next up. Oh yeah, so it is dehydration reaction. Dehydration, and then they didn't talk. Oh, they talk about hydrolysis here. So I jumped ahead. Okay. So oh, they showed the electron pushing here. I should have just scrolled down. Sorry. So basically, Amide, yeah, and a peptide bond is an amide bond. That's also very important to know. An amide bond is this bond right here, basically, this C and the N. And so the other thing that's important to see here is they call this thing partial double bond character. What this means is, so this here is a single bond between the C and C, right? That means it can spin freely. There is nothing holding it back from spinning. So this whole thing can spin this way freely. And then this part can do whatever it wants to spin as well. I don't know if that makes sense because it's not in 3D, but basically you can think of this part here. You can think of it like a four-wheel drive, right? Like if this is a car, right? And then this is the axle here, right? And then this is a wheel. This is a terrible picture, but I hope I'm getting across. If it's a four-wheel drive, then the wheels can spin in whatever direction they want independently. That's basically what it is here. The carbons can spin whatever direction they want independently. Now, that's not the case if it's a double bond. If it's a double bond, it's a little more locked down. It can't spin, right? But the thing is, this is not a full-on double bond. It's a partial double bond. Why is that? Because it has these two resonance structures. Sometimes the CN is a double bond, and sometimes it's a single bond when the electrons move up onto the oxygen. So that is basically just, it's basically, you're supposed to know that it's like not quite a double bond, but it's partially a double bond. So it doesn't have the full flexibility of a single bond, but it doesn't have the full rigidity of a double bond. Does that make sense? Okay, so, and again, if anything is confusing, just post in the chat and I will re-explain it. So now we talked briefly about hydrolysis, where a water molecule comes in and causes it to split up. This mechanism is quite, looks really long, but basically it just comes in and the electrons push in such a way that it splits up right here. And here you can see the two different amino acids. Protein structure. So now we get to an actual protein. Now we talked about the individual units, and now we have an actual protein. So a protein is just a really, really long chain. What's the end product of hydrolysis? It is 2 amino acids, and there's no water because the water is that actually, thank you for bringing that up. Where is the water now after we do hydrolysis? The water is actually now a part of the amino acids. So here is one part of the water, O minus here, and the other part of the water is okay, I see what they did. So so basically one of the protons is here because originally, so this NH is this one. I hope you can see. So basically, originally this only had one H on it, right? But now it has 3 H's on it, right? So, and this carbon originally had no oxygen on it, but now it has an oxygen on it. So that's where our 2 H's and our O end up. Now, they're not necessarily like that exact molecule of waters like H and O. It is that molecule of waters O, but it's not necessarily the protons, because the protons are always like all over the place. But that's a you don't need to worry about that semantics, right? Basically, this is where the water has ended up. The O is over here, and the 2 hydrogens are attached to the N. Does that make sense? Okay, cool. Good question. Good question. So now we go on to protein structure. So like I was saying, a protein is just a really, really, really long chain of these amino acids. So again, we can draw out the first part. So we have this is called the N terminus because it's the amino end, right? And this goes, and where does the amino acid end? Right here, between the C and the N. Because this N is the start of a new amino acid, right? Just like this N. So this N is another one, all the way until the next CN, the next peptide bond. And then this one all the way until the next CN bond. Okay, and so on and so forth until we hit the very last one, which is called the C terminus because it ends on the carboxyl group. And you'll notice that they have these blue parentheses N here. Blue parentheses N just means that, you know, repeat N number of times. So if it said like 5, that means that this is happening 5 times. So if this N said, if N was equal to 5 here, and there are 2 amino acids in the parentheses, how many total amino acids would there be in the middle there? So just to repeat that, if this N was 5, yeah, 10, exactly. There would be 10 in the middle. How many amino acids would there be in the entire chain? Considering the beginning and the end. 12, exactly. So the whole thing is N plus 2 because it counts the N terminus and the C terminus. And the N, usually it could be 1, it could be 2, it could be 3, but in any way, like that's just a math thing that you can solve. Any questions about that, about this polypeptide chain? Okay, great. So now, boop, 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 boop. So the N terminus refers to the side of the string. C terminus, yeah, we talked about that. Primary structure. Okay, so structure. Okay, so structure is super important. Protein structure is very, very important. So the first level of protein structure is its primary structure. And primary structure simply refers to the string of amino acids and is defined solely by the identity of the amino acids. This simply means primary structure is simply the order and the identity of the amino acids. That's it, nothing else. So it's talking about peptide bonds and the order of the peptide bonds and the amino acids. That's it. Primary structure is the simplest one. Don't overcomplicate it. It's just what amino acids are we using. By identity, I mean, is this an arginine? Is this a histidine? Is this, that's what I mean, yes. So the order and which amino acids they chose. That's what primary structure is. Now we get a little more complicated. Secondary structure is formed through hydrogen bonding interactions between the atoms forming the backbone of the protein chain, rather than interactions between the side chains. So what does that mean? Each amino acid has the NH group and the CO, the carbonyl group, right? These guys can engage in hydrogen bonding because this is an H and this is an O and this is an N, right? And these guys can do hydrogen bonding. I believe it's FON that can do hydrogen bonding and nothing else. So the backbone itself will interact with other amino acids in the whole polypeptide chain. So look here at this N terminus, this N, this H, right, is hydrogen bonding to this O, this carbonyl group of another amino acid down the chain. And this happens a lot. So basically this is what you're going to see: a carbonyl and a nitrogen. This is an amino group, right? Yeah, a carbonyl and an amino group hydrogen bonding. A carbonyl and an amino group hydrogen bonding. A carbonyl and an amino group hydrogen bonding. This is what we call secondary structure. Now, do not get confused with tertiary structure, which is when we use the R chains, when we use the R groups to do binding. So if we look at the R groups here, like this O negative can definitely bind to something. This OH can definitely get phosphorylated or something, right? These guys, these blue parts, these side chains can definitely interact, but that is not secondary structure. That is not secondary structure. It's only secondary structure if we're looking at everything besides the R group. If this guy is interacting or this guy is interacting with another amino acid, then it is secondary. But if the R group is interacting, then we're in the realm of tertiary. So wait, no, we're not done yet. Only when amino or carboxylic acid attaches to something, basically, yeah. Only when this part or this part attaches to something, or this. I don't think this one, this hydrogen, I don't believe participates in hydrogen bonding because this is a CH. So this NH and this CO, when they are bonding to each other, this is, and also this, another thing to note, it's not that this one and this one are bonding, because this is within one amino acid. That's not how it works. That's not how it's not physically possible. What it is that this one would be bonding over here, something like that. And that's what causes the whole chain to twist and turn into those crazy protein structures that we see. So that is that's secondary structure. And secondary structure gets a little bit more complicated than that because we have these patterns that we see. So again, secondary structure is composed of the hydrogen bonding interaction between the H of the NH of one amino acid and the carbonyl oxygen of another amino acid. So yes, Lamisha, that's correct, just between these two. Now there are two main structures or patterns or motifs of secondary structure, and that is the alpha helix and the beta sheet. And the alpha helix is this kind of spinny structure that we see. And again, notice that R groups do not contribute to the hydrogen bonding forming the alpha helix. So if they ask, alpha helix, beta sheet, what participates in it? It's not the R groups. It's only the backbone interactions. These are what we call the backbone of the peptide, of the polypeptide chain. These are called the backbone because the R groups stick out. You see how the R groups are sticking out? Whereas this is the backbone. So alpha helices, what part is not the backbone? The R groups. The R groups are not the back. The R groups, meaning the unique identifiers, like the OH or these long chains. Anything in blue here are not the backbone, and these parts that are not highlighted are the backbone. Yeah, this is another great point of structure and function, right? Many transmembrane proteins use alpha helices that span the entire membrane to transport ions from outside to inside the protein. So this is basically just a hint at when we study cell structure more in depth, but basically you would find alpha helices in transmembrane proteins. That's where they tend to show up. Now, the other major version of secondary structure is called a beta sheet or a beta pleated sheet. And what we see there is, again, remember, it's still amino to carbonyl, NH to O to CO, but the structure is a little different. It's not a helix, it's not a twisting helix, but instead it's like a sheet. And basically they kind of stack up in lines. So let me see if I can draw the line here in a way that makes sense. So yeah, so basically here's the line, here's a chain, 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 and then it kind of twists in on itself. It's kind of hard to visualize, but basically this long bond here is showing like that the chain continues to twist in on itself. And then we have the anti-parallel. So there are two different versions. There's one called parallel and there's one called anti-parallel. And the difference is that you'll notice it's very slight. It's a very slight difference. But the anti-parallel one, this is the beta sheet. These are both beta pleated sheets. The anti-parallel one has these straight hydrogen bonds, whereas the parallel one has these slightly off, like angled bonds. Why? Because the structure here is in parallel. So what parallel and anti-parallel is referring to is the amino acids themselves. Notice how in the parallel one, the amino acid is in parallel. Do you see how they're in parallel? How the carbonyl and the amino groups are in the same spot in the parallel version? But in the anti-parallel version, they are off by 1. They're offset by 1. So the O is one behind. The O is up here, but it's down here. And the N is here, but the N is up H here. So it's a matter of, are they on the same, are they 1-to-1, or are they 1 to like, are they, I'm not sure how to explain it in numbers. Maybe, let me see if this makes sense. If it's like, inverse is sort, yeah, I guess they are inverted. Yeah, that makes sense. They are inverted. But basically, the point here is that there are two different kinds of beta pleated sheets depending on the configuration. And the configuration happens when the chain is going this way and then flips in on itself and then keeps going up and then flips and then keeps going up. Or it can go down and then up and then down and then up. That's basically the two different versions of beta pleated sheets that you get. And that's what causes the slight difference. But overall, they're not going to ask you like, what's the difference in function between antiparallel and parallel, because that's not really something that you could tell. It's more so that it's just like a unique property of secondary. And they also say pleated because it ends up like stacking on itself like this. So this is just something to be aware of, just because they will bring up alpha helices and beta pleated sheets. But I don't think you need much more detail than this for this part. Any questions about secondary structure? Okay, then we shall move on to tertiary structure. So tertiary structure refers to structure that arises from interactions between the side chains, the R groups that we were talking about. So what can happen? Positively charged side chains can interact with negatively charged side chains, and then we get an ionic bond, right? And that's really strong. Polar side chains can be attracted to other polar side chains. Non-polar side chains can attract to other non-polar side chains through like hydrophobic interactions. So what does that mean? This is actually very important for understanding like what do I mean? For understanding proteins, protein hydrophobic. Maybe this will tell. For understanding why proteins arrange the way that they do. Yes. So notice here we have on the left an isolated protein. That means that this protein is not in an aqueous environment. It's just chilling by itself and is unfolded. Now, if you put the protein in an aqueous solution, which of the regions, when they say region, they mean like an amino acid, right? Or like a cluster of amino acids. Which of the side chains? Side chains are the R groups. Side chain is another word for R group. So R group, and side chain are the same terminology. So an R group, this thing, these guys, the R groups. You can call them R groups or you can call them side chains. They're called side chains because they stick out of the side. Like, so this is the backbone, and then the R groups here are sticking out of the side, if you can see it that way. So what was I talking about? Something. Oh, tertiary structure, right? So yes, this. Okay. So the question is, if you have a hydrophilic amino acid, do you think it wants to be out in the aqueous solution, or do you think it wants? Is it happy if it's in the aqueous solution or not? A hydrophilic. So hydro is water, and philic means it's the opposite. Phobia is scared of water because phobia is fear. So phobic is scared and philic is loving. So philic loves, and phobic, as in phobia, means like hates or fears, right? Philic loves and phobic fears. So if we have something hydrophilic, it's going to be happy out in the water. If we have something hydrophobic, afraid, it's going to hide away in the center. So when the protein arranges itself, the hydrophobic the regions, the hydrophobic amino acids, are all going to bunch together in the center away from the water. And it's actually kind of cool. Like all naturally, just because it's the best way for it to fold, the hydrophobic, this is a nice picture. Oh no, I just want the picture. So right here in purple, they showed the hydrophilic groups, and yellow, they showed the hydrophobic groups. So the hydrophobic groups don't want to be out where the water is, so they will fold up in such a way that they're all near each other. And they don't mind being near each other because they're all nonpolar there. Whereas the purple ones, the hydrophilic ones, will stick out with the water, and that's where they're happy because they like water and they interact well with water, because these are like negatively or positively charged, whereas the hydrophobic ones are nonpolar and uncharged. They can't deal with the partial charge on water. So that is basically to explain how tertiary structure works, how nonpolar binding works and polar binding works. And finally, we have something called a disulfide bond or a disulfide bridge. And this is just a very unique kind of bond that you see only in, this is a very unique bond that you see, and it happens between cysteines. So where's a good picture? This is too confusing. This is a nice picture. So a cysteine has an S at the end of it. Let's show back. Which one was polar? What's your question? I don't get it. Which what was polar? Oh, the hydrophilic molecules are polar because water is polar. So like attracts, like wants to be with like. So the hydrophilic molecules are polar. The hydrophilic amino acids have a polar group, and water itself is polar, so they're together. And then disulfide bonds are these guys. The cysteine has an S at the end, and the S and the S can link up and they form a polar covalent bond. So this is also very important. This shows up later with a lab test. Reducing and non-reducing. I'm not going to get into it right now, but this is just a very unique thing about cysteines. If you see two cysteines in like a protein chain, then you're like, oh, there could be a disulfide bridge forming here. And it's just you could see the cysteines here are linking together and causing a strong covalent bond here. Okay, so now finally we have quaternary structure. Quaternary structure is when we have multiple protein chains that start interacting with each other. So I think a very common one is heme. I think hemoglobin, quaternary structure. Whoops. Actually, no, this picture shows you right here perfectly, because it has two. You see, there's a blue and there's red. There's two blue ones and there's two red ones. So these are all four different subunits of well, it's two of hemoglobin A and two of hemoglobin B, I think, or something like that. And basically each structure, it's not just one chain, it's four chains all coming together and working together. And that's hemoglobin. So this is what we call quaternary structure. This is when individual strands are interacting with each other. So, okay, so that's basically, in a nutshell, the four different levels of protein structure. And you'll often find covalent disulfide bonds formed between two cysteine residues contributing to quaternary structure, but it doesn't mean that it's quaternary. It can also be tertiary structure. Cysteine residues start as tertiary structure. Hemoglobin is 4 hemes. Yeah, I believe so. Should be four heme groups. Yeah, heme group. Wait, no, am I wrong? Heme group. What is a heme group? Heme group. What is that? particular, is it just called a subunit? They're calling them beta chains here. Oh no, okay. I don't think it's a heme then. I think the heme is the iron thing. I think they're just chains. Don't remember exactly what a heme is, but it is, I'm sure, like it's the thing that, but I don't want to get, this is going to take a while to get through, so we'll come back to this. Let's go to modifications. So this is another thing that you may be asked. So we kind of touched on this. These are basically things that can happen to the protein after it's produced, after it's been fully set up. Phosphorylation. Phosphorylation can activate and inactivate a protein. This happens at serine, threonine, and tyrosine, which I showed earlier. Formation of disulfide bonds between cysteine residues. This can happen, you know, like with between other like subunits and stuff, right? Glycosylation is when a carbohydrate or a sugar gets attached to the protein. Ubiquination is when ubiquitin gets attached to the protein, which tells the cell to kill that protein and destroy it. So these are just 4 different versions of post-translational modification. Post-translation are just saying that the protein has been completely translated, and here are some additional modifications that can be made to it. So this is like, I'm sure there's an Anki card about this. Now, special amino acids. Glycine is special because it's not chiral. It has only one hydrogen. It has 2 hydrogens, meaning that one of the R groups is a hydrogen. And because of this, and because it's very small, it's very flexible. Because the hydrogen is not, you know, a very bulky atom, it's very flexible, and you would find it a lot in alpha helices that needs the flexibility. Cysteines are the ones that, you know, bind to other cysteines, and that lets them form disulfide bonds. And prolines are interesting. Prolines are very sterically rigid. So prolines are that way because they are nonpolar. Because look at this. This is interesting. The chain comes out here and then attaches back to this amino to the amino group. So this is the only one where, well, no, it's not the only one. Is it histidine or is it not? Histidine. No, histidine doesn't link back to it. So this is the only amino acid where it links back and makes this like rigid structure here. And that's preventing this, like the spinning that we were talking about over here. So that makes proline very rigid. And where do we want that rigidity? We see that rigidity on certain like turns of the alpha helix. Five-member ring that does not have a lot of rotational ability often creates a kink in the protein strand. Found in the regions a protein should be immobile or when a bend in the structure is needed. It's like a hard turn in the chain. So those are some unique amino acids. Stability. Okay, so we just were talking a little bit about hydrophobic and hydrophilic. So here we have these waters out, and the water is polar. And so for this protein to be happy, the parts of the protein that are interacting with the water should also be polar. But the inner parts of the water, of the protein, should be non-polar because they don't want to be, you know, attracted to the water. They're just going to be attracted to themselves, and they're just going to be inside. Why is this good? This is good because attract each other. So the hydrophobic residues will be attracted to each other, while they will also be repelled by the polar aqueous environment. For this reason, hydrophobic amino acids will be found in the interior of the protein rather than the outside of the protein. This can also be explained from a thermodynamic standpoint. The hydrophobic effect also has to do with entropy. So entropy is disorder, like how messed up things are, right? If what do you think is more disordered? Something that's very rigid or something that is non-rigid? If something is actually non-rigid, right? Yes. So excellent. So if the water is able to interact with this polar molecule, or if it's stuck against non-polar molecules, do you think it's more, in which case does it have more freedom? If it's next to polar molecules or if it's next to non-polar molecules?
I'm sorry, what are you referring to?
So I'm saying, basically, these water molecules are not just still like they are in this picture, right? They're always moving around and they're floating around, right? When would they have the most freedom to float around? If they are near a polar molecule or if they're near a non-polar molecule? A polar molecule, yes. And I'll show you a very nice diagram that explains this really well. I think it's called solvation layer, solvation shell. Yes, okay. So this is solvation shell. Water molecules interact unfavorably with hydrophobic sections. So water molecules are forced to lock their rotation and form shells around the protein. So here, if this is hydrophobic, look at how the water molecules have to arrange themselves. They have to they get locked in this rigid conformation because they can't insert themselves into this, into this water. They can't really interact with the hydrophobic molecule. So they're just stuck this way. However, with hydrophilic molecules, they can move around willy-nilly as much as they like. So in this case, they're showing like separate molecules, but basically the idea is the same for this. The water molecules can move around on the surface of the protein as much as they want, unlike if they were hydrophobic. If it was hydrophobic, then they wouldn't have the freedom to move around. They would kind of just be stuck there, forming a solvation shell, isolating the hydrophobic proteins. So that is like the entropic explanation for why this happens. Because if it's set up this way, if it's set up with hydrophilic on the outside, it gives the water molecules the most disorder, the most freedom to move around and be in whatever, orientation they want to be. That's a little bit complicated. Does that make sense? Or is there any question on this? I don't know if it makes sense or it doesn't make sense. What was the original answer? What was my original question? I think my original question was, would water molecules be happy with if they were in the presence of a polar thing, of non-rigid for polar or non-polar? So yeah, would they be rigid? They would be rigid. So the answer is water molecules would be rigid in the presence of a hydrophobic molecule. Water molecules would be non-rigid in the presence of hydrophilic molecules. You can think of it as like, like philic, like they like each other, so they're happy to move around however they want. Phobic, they're afraid of each other, so they're not that comfortable. They're kind of standoffish. If you want to like personalize it that way, you can think of it that way. But entropically, because they're like moving around and there's like, it's a party here. They're all moving around. They're happy to move around whatever way they want. Disorder increases. There's more movement in the water molecules and there's more microstates. So non-rigid for polar. Correct. If it's a polar molecule, the water molecules are non-rigid. If it's a non-polar molecule, if it's a hydrophobic molecule, the water molecules have to be rigid to accommodate this structure. H2O molecules form a rigid hydrogen bond network around aggregated hydrophobic groups, whereas H2O molecules form hydrogen bonds with hydrophilic groups. Because they can't bond to this guy, they have to kind of like cage it in, whereas they can bond to the hydrophilic molecules, so they're happy to like link and then unlink and whatever. They're very go with the flow. Okie dokes. So I think that's pretty good. And yes. So yeah, if the outer layer of the protein is comprised of mainly hydrophobic residues, the number of hydrogen bonds that the solvation layer can form, the solvation layer being the layer of water that's in contact with the protein. So if the outer layer is hydrophobic, the number of hydrogen bonds that the solvation layer can form becomes extremely limited, right? And that's exactly what we see here. The water can only bond to itself in a very limited way. It can't freely bond wherever it likes. Notice, look at this oxygen right here in the corner. It's not getting to bond to this hydrogen here in the corner. It's not getting to bond to anything. Whereas these guys are all maximally bonded as much as they want. So yeah, so the number of hydrogen bonds that the solvation layer can form becomes limited. This requires the water molecules to be oriented more precisely / rigidly, resulting in an unfavorable reduction in entropy, a reduction in disorder, right? And that's what we see here. This has become more ordered, and this is less ordered. Does that make sense? That was like a brief tangent into entropy and chemistry, but again, you're going to see as we go through all this stuff, everything is connected. So the more you learn here, the more you're learning a little bit about chemistry. When you learn chemistry, you'll learn a little bit more about biology and so on and so forth. That's how it is. Okay, good. So now, protein, what's the opposite of entropy? Order? There's just disorder and order. I don't know what the opposite of entropy is. Enthalpy? No, enthalpy is not the opposite. Enthalpy is related to heat. Enthalpy is a different concept. It just sounds similar. So protein folding, the solvation environment of a polypeptide chain directly influences the way a protein folds, right? I think we understand that very clearly, right? The protein is going to fold based on what maximizes the water's entropy. The water wants to be happy, the protein wants to be happy, it's going to fold in that way. So protein folding is, how the protein folds, and denaturing refers to unfolding a protein. So when you crack an egg, you notice that it's like kind of liquidy, right? But when you cook the egg, it turns into this like white rubbery thing, right? You are denaturing the proteins in the egg through heat, through high temperature. You've denatured a protein. So that's why my mom loves to say cooking is chemistry, right? And that is what it is. When you're cooking an egg, you're denaturing the proteins of the egg and there's multiple ways that you can denature proteins, either through high temperature, which disrupts the hydrogen bonding, or high salt concentration, which also messes with the hydrogen bonding and also stuff like a disulfide bridge or the bonds between the negatively and positively charged amino acid groups, or high and low pH, because as we briefly talked about before, amino acid groups can get protonated and deprotonated, right? And if they're protonated and deprotonated, that affects how they're bonding with other amino acids. So these are a couple of different ways that we can have denaturing occur. So now, these environmental changes can result in protein denaturation. As proteins form, is highly related to function. Protein denaturation. Yes, so the structure, a protein form is highly related to its function. So denaturation usually means a loss of function. And sometimes the denaturation is reversible. Sometimes it's irreversible. For example, if you cook an egg, you can't, really turn it back into an uncooked egg. But if you transfer I'm not sure about an egg, but in some cases, a protein in one environment can be, if it was in a high pH environment, and then you lowered the pH by adding some acid, you could feasibly turn it back into a functional protein, just because you change the environment that the folding that encourages the folding. So overall, many proteins cannot refold to the native state once unfolded. But in biological systems, there are chaperone proteins that help folding proteins back together. So overall, this is pretty much everything that you need to know about amino acids. And again, the part that we can't cover that you have to learn on your own is memorizing the amino acids themselves. That's all of the stuff about amino acids you should know. And yeah, there's like a little glossary here, which is kind of nice. But I think we have all of that in our Anki deck already. Pretty sure. Protein structure. Yeah, see, so this is the exact same thing. Denaturation, high temperature, high or low pH, high salt concentration, and I've added one, urea. Urea is also something that affects, oh, and I have a nice diagram here. Okay, this is also really important. Yes, I wanted to mention this, right? So as we go through the content like this, right? How are you going to remember it, right? You're going to remember it with Anki. And Michaela, I'm sure you're kind of confused what this is if you haven't used it before. I will explain it a little more in depth. Basically, these are flashcards, right? And so okay, perfect. So these are there's a video talking about Anki and stuff. But for a brief intro, we have these flashcards. This is the deck that I made when I was studying, and these are, so when we study this material, right, we studied about amino acids, right? So now what we're going to do is open up all of these cards about amino acids, basically. So this is amino acids right here, right? So charge on amino acids, peptide bond formation, peptide bond hydrolysis, denaturation, carbohydrate nomenclature is not it, right? So these are basically like the 4 amino acid cards that we want to, where is it? Unsuspend. I want to unsuspend it. Oh, toggle suspend. There we go. So now that I've toggled the suspend, now these cards are open for me to read about. And let me just search for amino acid. Where is my amino acid card? Oh, here it is. Amino acids. So this has like some random stuff about amino acids, basically. And you're going to want to unsuspend that. And then this is like a giant card that has all the amino acids. This is not the best way to memorize it. This is just like a giant card, but you're going to have to memorize the amino acids like one by one, or like, you know, break it down into separate bits. Okay, so that's basically the idea there. As we go through the content, right, we're going to open up the cards and we're going to go through the cards one by one. Okay, so now the question is, right, hold on, let me just Okay, so now we're on non-enzymatic protein function, and non-enzymatic protein function is basically like proteins that don't act in an enzymatic way. That's just the same thing. But enzymes basically catalyze reactions, right? But there are also proteins that are not catalyzing reactions, such as structural proteins, right? And actin is one of those structural proteins. So there's a lot of structural proteins in our cells, right? Proteins that are just there to give our cells structure and shape. So we have, you know, the cytoskeleton and the extracellular matrix, and these guys are, we have your actin, tubulin, collagen, elastin, and keratin. You can, that's just something you can put on an Anki card and like memorize if you want. Actin and tubulin are primarily found within the cytoskeleton of the cell, while collagen, elastin, and keratin make up the extracellular matrix. So again, that's just another fact, right? Actin is the most abundant protein in eukaryotic cells, and it turns into these long microfilaments, which are also called actin filaments. And actin filaments have polarity in the sense that they have they're not equal. There's a negative end and a positive end. Oh, like a magnet. I like that. Yeah, just like a magnet. A negative end and a positive end. And or a plus and a minus end. The plus end of the microfilament is where new actin monomer is attached to a polymer. So again, this is another polymer, right? Actin is a monomer and the whole thing is a polymer. The plus end is where the new actin is getting added, and the minus end is where they dissociate. And this is done through ATP, right? And we have this thing called treadmilling, which is actually very fascinating. Actin treadmilling GIF. Yes. This is actually kind of pretty cool. So it can move like this by dissociating the back part and adding to the front part. So the actin can contribute to structure in this way and move around. So yeah, results in an apparent motion of actin strands across the cytoplasm, which is often useful in cell movement. So if you ever thought about phagocytosis or a cell moving, it's moving this way by like this thing like pushing out into the membrane and like kind of pushing the cell forward. It's like if you imagine like if we actually this is kind of true, like our skeleton, if our skeleton was pushing us around and like we were kind of like just blobs of meat and like our skeleton was pushing us around, that's essentially like what's happening here. So actin, okay, so then the other thing that's really important is myosin. Myosin is what we see in muscle cells that helps us contract muscles and have movement. So this is a very in-depth cycle that you need to know pretty well, and so we're going to go through it. Essentially, and also let me get my card here because I think my card here is pretty good. Contractile cycle. Yes, okay. So here, essentially, what happens in muscle cells is we have, in the normal state, it's kind of like this on the left. So then this little pink thing sticking out is the head of a myosin. And it's attached to a phosphate and an ADP molecule. When calcium comes in, it acts on, so this part, this is like an actin filament, and this is a myosin filament. And when this happens, when calcium comes in and opens up the actin filament, it allows the myosin to grab on to the myosin binding site. Yes, that's what it's called. And then when these guys release, the myosin does this thing called the power stroke, which pulls on the filament. When it pulls on the filament, that is essentially muscle contraction, just a very small amount. But this cycle happens rapidly. Then the ATP comes, and when the ATP dissociates, it goes back to its normal state. But as long as the calcium is there and the myosin binding sites are open, it will repeat the cycle. And that's essentially what's happening inside your muscle cells repeatedly when you contract your muscle. This power stroke cycle or contractile cycle. So back to here. So that's basically what this diagram is showing, the same thing, where it's pulling, you can't see the pulling that well here, but overall, the whole thing is getting pulled closer and closer. So let me see if I can find a good picture of the pulling. No, maybe, no. Okay, I can't find a very good, but basically the idea is that when this happens repeatedly, it gets pulled until you can't pull anymore and you've contracted fully. So many myosin heads must work together at once to produce enough force to contract a muscle. This is just showing a very single actin microfilament, but this happens in bundles, which is to say it happens like, I know I have the picture, I just can't, I'm just not sure where it is. Oh yeah, sort of like this. These are little bundles of these microfilaments that are pulling and contracting. So and cross bridge cycle, yes. So this whole cycle, the way I kind of described it, you need to know each step of it. At least it's something that they can ask you about. this cycle of power stroke and then recocking the myosin head and then going forward and forward until the calcium dissipates and there's no more need to contract. So any questions about myosin? Any questions about this? And this is, you're going to revisit this when you study like muscles and stuff. That's gonna definitely come up again. Okay, so then we also have cytoskeletal elements, and I'm pretty sure I have a really nice card about this. Yes, so the cytoskeleton, right? The cytoskeleton is composed of protein fibers that help with cellular movement and maintaining the cell structure and shape. And there's three different things. There's microfilaments, microtubules, and intermediate filaments. And there's all these, interesting properties and functions of these guys. Microfilaments are the ones that help you with muscle contraction and help you with movement, because we saw that treadmilling, right? That ability of the negative end to dissociate and the positive end to reassociate. Microtubules are these large hollow tubes that help for transport, for vesicle transport. And they also can help with, transport. And there's also kinesin and dynin. I actually don't know how to pronounce these properly, but these guys are that funny little GIF that you might have seen. This thing, where it looks like it's just walking along. So what we're seeing here is a kinesin or a dynin holding a vesicle and transporting it along the green microtubule track on the bottom here. And so this is really quite cool because these guys can walk along the track and transport stuff. It's pretty cool looking. Just kind of one step at a time, just keeps trucking along and it transports whatever it needs to transport. So yeah, I agree. Pretty cool. When I first saw that, I was like, wow, that's super interesting. That's very neat. So kinesin and dynin, the distinction between them is that the kinesin goes away from towards the periphery of the cell, whereas dynin takes stuff towards the center of the cell. That's basically what it is. That's, again, a lot of these are things that are just facts that you just stick in an Anki card and you memorize, and that's all it is. Intermediate filaments are, in between, and they're mainly found in desmosomes. That's really the main thing that you would memorize about intermediate filaments. And they're made of stuff like keratin, whereas microtubulars are fortunately made of tubulin, very simple. Microfilaments, aka actin filaments, are made of actin. And the way another handy little mnemonic, actin sounds like thin. These are the thin ones. These are the thin filaments. Whereas microtubules are big hollow tubes. And then intermediate is just intermediate, in between. They're not super thick, they're not super thin. And we have some nice pictures of the cytoskeleton here, microtubule being hollow here, and so on. Okay, so what else? Microtubulin. Oh, and then they talked about kinesin and dynin here, which we already talked about, the motor proteins, how they move. The kinesin moves away and the dynin moves towards the cell. And they talk a little bit more about collagen and elastin and keratin. Collagen is a helical fiber made of 3 interwoven strands. It's in the extracellular matrix. Collagen provides structure to our tissues, bone, ligament, and tendons, right? Like, I'm sure collagen in your nose and your ears, elastin cross-linked structure of the extracellular matrix, again in the extracellular matrix, and keratin, is in our nails and our hair. So these are the proteins that are majorly found in our body, but they're not enzymes. They don't act on anything. They're just there to provide structure. Transmembrane proteins. Okay, so GPCRs. So G protein coupled receptors. Interesting. So G protein coupled receptors, they're not really going into depth in it here, but I wonder how in-depth you need to go for GPCRs, because I think we have it pretty in-depth here. Oh, okay, sure. So GPCR, it's not here, is it? G protein. I wish I just look up a G protein. But basically, G proteins are these things that GPCR, G protein coupled receptors are very, very important in cell signaling. They are a very, very common style of signaling proteins. And where did it say it? You may also see GPCRs referred to as 7 transmembrane receptors as they span the cell membrane with 7 different hydrophobic regions. So that's what we see in this picture here. You see this barrel, 1, 2, 3, 4, 5, 6, 7. So this is a GPCR. And when a GPCR receives a signaling molecule from the outside, it will trigger a signal cascade on the inside of the cell. And that's going to tell the cell a lot about it's going to evoke a response in the cell. And the actual way that it works is that let me see if I can find a clearer one. GPCR cycle. These pictures are not that clear. Then again, I'm not even sure if you need to know the cycle in depth. You learn it in biochemistry, but I don't know if they want you to know it for this. Okay, so I mean, this is kind of a decent one. Yeah, this is a decent one. So this is a G protein coupled receptor. If the protein gets a signaling molecule, what happens is that it's called a G protein coupled receptor because it's got this GDP. GDP is, so you've heard of ATP, which is adenosine triphosphate. GDP is guanosine triphosphate, I believe. So GTP will come in and replace the GDP, and that's like the go signal. Once we get the signaling molecule, a GTP comes in and replaces the GDP at the alpha subunit. That's the go signal. That causes the alpha subunit to separate from the beta and the gamma subunit. And then while this is separated, a bunch of signaling stuff happens downstream. And what kind of signaling depends on the kind of G protein. But a bunch of signaling happens. And then whenever the signaling stops, the GTP is going to be dephosphorylated and the P will leave. Once the P leaves, it's back to the inactive GDP. When that happens, they refuse, the alpha, beta, and gamma come back together, and then there's no more signaling. So that's essentially how a G protein coupled receptor works. I was sure that I had a card about G proteins though. I guess not. Maybe I took it off because, oh, no, wait, I did. Did I? Yes, this is a G protein. Yeah, I did. Oh, I specifically called it phospholipase C. So this is a particular type of G protein. Basically, so again, we see the same exact structure. The main structure is the same. It starts as a GDP with an alpha, beta, and a gamma. Once the signaling ligand attaches, it causes the GDP to be replaced with a GTP. And that causes the signaling thing where the alpha separates from the beta and the gamma. While the alpha and the beta and the gamma are separated, we have this whole signaling pathway happening. And this signaling pathway is unique for the type of GTP. In this case, it's something with, releasing calcium from the ER. And then once that ligand is gone, the GTP will be replaced with a GDP and we'll go back to no more signaling. So we can see that the signaling molecules being produced here are this IP3, which is going here and causing calcium to be released. And once calcium is released, you have activation of protein kinase C, which leads to phosphorylation of proteins, which leads to some kind of function. So that's what a G protein cascade or signaling molecule looks like. I'm not sure how in depth G protein should be, but I will check and let you know. But now you know. Pores, ion channels, and active transporters. We talked about this a little bit last time. Pores and ion channels are passive transport. Molecules go down their concentration gradient. So they go from high to low. Aquaporin is very important. Aquaporin is the pore for water. So water will just flow through these ion channels, right? Active transporters are different. Active transporters move ions and large molecules against the concentration gradient. They don't allow things to go from high to low, but rather they move them from low to high. So I'm sure I can show you transport. Yeah, so these molecules, if this was a simple diffusion situation, then the molecules with the high concentration would want to go up where there's lower concentration. But since this is an active transporter that's going against the concentration gradient, they're moving the small amount of molecules to where there's a high amount of molecules. We're going opposite. That's basically what this is. Active and transport. And so these guys are proteins that facilitate the transport of ions and such. And signaling proteins are basically hormones. So hormones are very important, especially when you cover endocrine system and anatomy. Hormones are slow-acting chemical messengers that travel through the bloodstream. Okay, and hormones. So we have different types of hormones. Peptide hormones are molecules that look like this, and they're based on they are protein-based hormones essentially. There's also like cholesterol-based hormones and stuff, but this is a protein-based hormone that binds to cells and changes their receptor, causes changes on the cell basically. Peptide hormones are particularly important in HPG axis. This is more towards anatomy. I don't think we need to really cover this right now, because this is kind of how hormones influence the system. Yeah. Antibodies are obviously another kind of protein. I guess they're not enzymatic. Yeah, they're not really enzymatic. They just exist to bind to antigens, right? That are foreign, and antibodies help neutralize things through three different ways. This is definitely in the Anki deck. They agglutinate them, right? Like once they attach to a virus or to a bacteria, right? They attach and prevent that virus or bacteria from moving around freely, and that's agglutination. Neutralizing, I'm not sure what neutralizing is. Maybe it's like it prevents its let's see, opsonization. Oh, opsonization, I don't have a glut or antibody. Antibodies, yeah. And the structure of the antibody is also important. Just that it looks like this Y shape, right? But it only binds to the antigen at the two ends here. So the two ends, this end here of the Y is called the variable region, whereas the rest of it is called the constant region, because the purple part here, the constant region, is not undergoing any changes to bind to the antigen, whereas the variable region is highly variable and changes so that it can match antigens. Yes. Oh yeah, opsonization. I forgot what that means. Opsonization. More susceptible to phagocytosis. Okay, so when it binds to a pathogen, it makes it easier for other cells to phagocytose it and eat it, basically. So antibodies, yes. And that's about it. So these are all the proteins that are not enzymatic in our system. Any question about these guys? There's a bunch of them. They're just like random proteins to know about. But these are all going to come up more when you do anatomy and stuff. Especially, these are very like anatomically linked. Okay. Good to go for enzymes. So enzymes. So perfect transition. So enzymes work to catalyze a reaction. The non-enzymatic proteins that we just saw are not catalyzing anything. They're not making a reaction happen faster. They're just existing in structural ways. So enzymes actually have catalytic activity. So catalytic activity means they serve to make a reaction occur faster. So what does that mean? So catalytic activity, this is a nice example. I like that. So in this case, they're saying like, to go from A to B, if we think about A as the reaction and B as the product, right? If you walked, it would be slow, but if you took a car, it would be fast. And it would be the same thing, right? So, but one would be faster. The reaction, the beginning and the end is the exact same thing, but we get there faster. So they're saying as the distance is not changing and the destination is not changing, but the speed to get to the endpoint is faster. So a car is an enzyme. Or an enzyme is a car. An enzyme makes this process faster. So the three things that the three important parts of an enzyme. First things first, lowers the activation energy of a reaction. So what does that mean? The reason that these reactions are slow is because they take a lot of energy to build up, to hit the transition state where they will go into the products. What an enzyme will do is lower, so the dashed line is an uncatalyzed reaction, and the solid line is a catalyzed reaction. So if you don't have an enzyme, it may take a long time for an enzyme for the reaction to occur, because we need to hit a certain threshold before the reaction can occur. But the enzyme will reduce the amount of energy required for that threshold, and therefore it can make the reaction happen faster, aka catalyze it. Now, notice that where the reactions are in terms of the free energy, the reactants and the products are both the exact same, regardless of catalysis or not catalysis. That is extremely important. An enzyme does not change the reactants or the products in terms of their energy, in terms of what is produced, in terms of what the reactant is. Those all remain the same. The enzyme is only doing one thing, reducing the activation energy to speed up the timing of the reaction. That's it. It doesn't do anything else. And the enzyme is also not consumed in this reaction. All it does is just gives it, like the same way if you took a car, right? You wouldn't suddenly lose the car. The car is fine once you leave it, right? So that's the same thing. The enzyme is just a car. And the car is not changing the destination you're going to. The only change is speed, correct. The only change is speed, that's it. And the activation energy, because the activation energy was the barrier to the speed. So they're saying affects the kinetics of the reaction. When we say kinetics, we mean the time frame, but not the thermodynamics of the reaction or the equilibrium constant. That's just fancy saying for like, we don't affect the free energy. We don't affect the reactants or the product's beginning or ending point. Okay. So, an enzyme affects the kinetics of a reaction by speeding up the rate of the reaction. No, the reaction rate is the rate at which reactants are consumed or the rate at which products are made. Enzymes do not affect the thermodynamics or equilibrium constant of a reaction. So here they're giving us, so what is their, another example? We have a machine that converts red spheres into red cubes. Normally, if you give the machine 10 red spheres, it will produce 5 red cubes an hour. Now let's say a catalyst acts on the machine. If you give the machine 10 red spheres, it will produce, okay, this is kind of like a weird example, but they're basically just saying it happens faster in very convoluted terms. Okay. Enzymes are regenerated because they don't need to be consumed. They are just there to help the reaction occur. Enzymes often end with ASE. I'm sure you've noted like polymerase or ATPase or helicase, whatever ase, right? Those are enzymes. Regeneration. I think they're just trying to show here like why it's important that enzymes don't get consumed, because then you just have one enzyme and you're good to go. You can constantly speed up that one cycle. It's like a machine that works continuously versus a machine that breaks down after one try. It works continuously and it doesn't get messed up when it does these enzymatic reactions. Okay, so now we get into kinetics. All right, so this is okay. Okay, so an enzyme attaches to a substrate, the substrate being whatever it's working on, like the reactant that it's working on to catalyze, right? And then it forms this ES, which is called the enzyme substrate complex. Once the reaction is complete, it will dissociate into the product. The substrate has turned into the product, and the enzyme itself is unchanged. It's just a normal enzyme that it was. So two ways that are commonly mentioned are the lock and key model and the induced fit model. So these were two different theories for how enzymes were working on substrates. The lock and key model was the idea that the enzyme perfectly fits the substrate, and that's how they lock together. Just like a lock and a key, right? It's self-explanatory. Whereas the induced fit model is a little different. They're saying that they're not exactly the same, but once they find each other, the enzyme will shift to lock onto it a little bit better. Sorry, not just the enzyme. Both of them will shift to hug each other a little bit tighter. And this is the more accepted model now, the induced fit model. It's now more understood that both of them kind of do a little bit in making those interactions tighter and sealing them together. So yes, both will undergo slight conformational changes to improve their binding to one another. That's a much better way to say it. So that's the two different models of enzyme substrate binding. Okay, so types of enzymes. Alrighty, so this is something good to know. There are 6 major types of enzymes. So the types of enzymes, and again, because there's many different reactions that can be catalyzed, right? So isomerase must deal with isomers, right? So an isomerase is going to, let's see if we can find an example that shows all of these types of enzymes. I want something that shows the examples. This looks good. Okay, so an isomerase is going to rearrange, molecules. So this is now, these have the same components, but in different structure, different order. So that's an isomerase. Then we have, what else do we have? A ligase. A ligase is very friendly. It just joins two things together. This is pretty nice. You often hear about this with DNA ligase, and we'll get to this when we talk about DNA and Okazaki fragments. Then we have transferase. A transferase is going to transfer a functional group from one molecule to another molecule. So if you look at a transferase here, you can see originally this has an A and a B, and a C is by itself. Then when the transferase does its action, The A is by itself because it transferred this yellow B to the other part. So that's transferase. Then we have a lyase. A lyase breaks a molecule without water versus a hydrolase, which breaks a molecule with water. And we actually saw hydrolysis earlier today when we talked about peptide bonds, right? So a hydrolase uses a water molecule to split up two components. And what it does is it gives a hydrogen on one end and it gives the OH on the other end. That's how a hydrolase works. Whereas a lyase will put things together without using water. These are kind of uncommon, but they show up once or twice, but it's just another type of enzyme. And the last one is oxidoreductase. This is a big one. This involves oxidation reduction reactions, which we'll talk about more in depth when we get to chemistry. That's basically where these little things are, basically electrons, right? And you're moving the electrons from one compound to another compound. So once something is being reduced and something is being oxidized. So reduction is gain of electrons, oxidation is loss of electrons. The mnemonic is oil rig, which stands for oxidation is loss, reduction is gain. So oxidation is loss of electrons, reduction is gain of electrons. And an oxidoreductase is catalyzing that transfer of electrons from one molecule to another. So those are the 6 major types of enzymes. You can definitely be asked, like, what kind of enzyme is this? That's something. What else? Cofactors, coenzymes. Oh, sorry, before we move on, any questions on the 6 different types of enzymes? That's okay? Okay, cool. Now we are on cofactors, coenzymes, and water-soluble vitamins. So this is a thing where, like, enzymes don't really act by themselves. Like, they often have something hanging out with them that helps them. So for example, if it's a coenzyme, it's a protein, right? If it's a cofactor, it's not a protein. It's like an ion or a vitamin or something. So cofactors can be like magnesium. That's a pretty common one. If a cofactor or coenzyme is bound extremely tightly, such that it's always there, it's called a prosthetic group. It's like a prosthetic for the enzyme that helps it function. And when they're attached, they're called hollow enzymes. I think of this as, my mnemonic for this is whole enzymes, because they're functioning correctly, because they're put together whole, like they're whole, right? Like nothing is missing. They're hollow enzymes or holo enzymes. And the opposite is apo enzymes, where they're missing their cofactor. Yes. So as we talked about, you need to know the 1-letter code and the 3-letter code and the structures for all the amino acids. They are literally guaranteed to show up on the MCAT. And interesting. So then their question here is arginine or valine more likely to be catalytically important? So the reason they're asking you this question is because arginine and valine have different properties. Valine is a nonpolar molecule, whereas arginine is a positively charged amino acid. So which of these, the positive or the nonpolar, which of these would be more important catalytically? And when I mean catalytically, which one would be more likely to be involved in a reaction? The one that's positively charged or the one that's nonpolar? What do we think? Positive charged or nonpolar? Which one would be more likely to be involved with a reaction? We've seen that reactions are all about, how do I explain? we've seen that hydrophobic stuff, right, doesn't really do well in the aqueous environment, right? So these guys that are nonpolar, the valine is nonpolar and hydrophobic. So this is not going to be in charge of, this is not going to be something that is very enzymatically active. No, not which is more common, which is going to be, which, so if we draw out a enzyme, right? Like let's just imagine like we draw a bunch of squigglies and this is a protein, right? And then the enzyme has an active site, right? Let's say like that this part right here is the active site. Let's say that this part right here is the active site that attaches to something, to its substrate, right? That active site, is it going to be hydrophobic or hydrophilic, do you think? Because it's outside, it's on the outside, it's facing the environment so that something can bump into it and attach. So if it's on the outside of a protein, it's going to be in contact with the water and the aqueous environment. So if it's on the outside of a protein, it's more likely to be hydrophilic, or it's more likely to be charged. Whereas the hydrophobic stuff wants to be on the inside. The hydrophobic stuff does not want to be in contact with the aqueous environment. So that's why they're saying between arginine and valine, oh, no, I should not mix up the colors. Between arginine and valine, valine is the hydrophobic and nonpolar, so it's more likely to be on the inside. Arginine is positively charged, so it's more likely to be interacting on the outside. So now you probably don't know these amino acids yet, but the idea of the properties is something that is something that should start to make sense, in that the positively charged and the polar amino acids are more happy to be on the surface or outside, and the non-polar uncharged amino acids want to be on the inside. Does that make sense? Okay, awesome. So, okay. And that is, again, this charge stuff and all this stuff is going to come up again when we discuss chemistry and organic chemistry and stuff. So you will continue to see this, basically. Okay, enzyme kinetics. Oh boy. So enzyme kinetics is a lot, actually. Enzyme kinetics is a topic unto itself. Let's see. Okay, let's see. Enzyme kinetics. Enzyme kinetics. So basically, actually, no, I can't even say basically because it's not even basic. Like I have to go step by step. So enzyme kinetics is the idea that, how can I explain this well? And they're basically giving everything, yeah, and then they're going to inhibition and stuff. Okay, so enzymes have different activity based on the substrate concentration. Okay, so let's think about it like this way. Let's say I have let's see here. Let's go for text. Let's say I have 5 enzymes, or let's use some other example. Rather than 5 enzymes, let's say I have I don't know. What's a good example? Let's say we're at Subway and we have 5 people, 5 sandwich makers. And we get 1 customer coming in. How many sandwiches are going to be made? If one customer only ever orders 1 sandwich, how many sandwiches are going to be made? 1 sandwich, right? 1 sandwich is made. How many sandwich makers does it take to make 1 sandwich? Again, it's just 1, right? It's just 1 sandwich maker making 1 sandwich. So therefore, 4 makers aren't doing anything, right? Not doing anything. This is basically, so now to link this back to like actual science, right? Or biology, the sandwich maker is the enzyme. It's the one that catalyzes a reaction. Whereas the customer is the substrate. The customer needs to be present for the reaction to actually happen. And the one sandwich being made is a very low rate of production, right? Because there's not enough substrate. So that's kind of what this graph is trying to show you. This graph is showing you that on the x-axis, we have the low end of substrate concentration and the high end of substrate concentration, right? At the low end of substrate concentration, we're not having that much enzyme activity because, and so Vmax is called, is the rate of the reaction velocity, right? So it's kind of like a measure of how fast are we getting this reaction happening, right? So if there's only one sandwich being made, we're not making that many sandwiches, right? We're not making as many sandwiches as we could be making. How many sandwiches could we be making under ideal conditions? What's the most amount of sandwiches that we could be making here? 5, right? Excellent. Yes, we could be making 5 sandwiches at most. When would we be making 5 sandwiches? Exactly. If there's 5 customers or 5 substrate, right? And then we produce 5 sandwiches produced, aka the product, right? So that's what we see here at the end, and that's what's called Vmax. This is the maximum velocity of our reaction. Why? Because what happens if we have 5 makers and we have 10 customers? How many sandwiches do we make? Still 5, right? That is why at Vmax, if you crank up the substrate concentration, you no longer increase the reaction velocity because you already have the most amount of enzymes that you have, and all the enzymes are occupied, so you can't make any more product. So this is what this graph is essentially telling you. At a very low substrate concentration, right here, we don't not all of our enzymes are occupied. So we can continue to speed up our reaction if we add more substrate, and more substrate binds to more enzymes, right? So we keep going. So like, let's say this is 1 customer, right? And then this is 2 customers. This is 3 customers. This is 4 customers. And then boom, this is 5 customers. If there's a 6th customer that comes in, it will not go up. That's why there's this little blue line here, because we already hit Vmax. We don't have 6 sandwich makers. We only have 5 sandwich makers, so we can only make 5 products. Now, over here, so what is our Vmax in this hypothetical? Our Vmax here is what? Our Vmax, so the Vmax is like the maximum product that we can make, right? So what is the maximum product that we can make? Five sandwiches, right? We can make 5 sandwiches is the maximum product. Now, what is half of our maximum? How many sandwiches would be at 2.5, right? Let's just ignore the fact that there can't be half a customer for now. Like, let's just imagine 2.5, right? 2.5 sandwiches is half of our max, right? So now, how many customers would there be if we were making 2.5 sandwiches? Yes, 2.5 customers, right? And that's what we call KM. KM is defined as the substrate at 1/2 Vmax. So if you know the Vmax, then you know half of Vmax. And if you know the substrate, if you know the half of Vmax, and you know the ratio of the substrate to the product, then you can know what the Km is. So Km is the substrate, is what we call the substrate concentration. The substrate concentration at 1/2 Vmax. So, any questions about this analogy, or 5 sandwiches produced? Oh, 5 sandwiches produced, not 10. Okay. Yeah, does this make sense? Okay. So, okay, good. So what we just saw here, this graph is called a Michaelis-Menten plot. And a Michaelis-Menten plot shows you, basically just plots this relationship. It's like how much substrate leads to how much reaction velocity. So now going forward, this is kind of blocking this. I want to move this. Let's stick it up here for now. So as they talk about here, the two big values are the Vmax and the Km, right? As we just described. So as the substrate concentration increases along the x-axis, the curve begins to flatten out as it reaches Vmax, because Vmax is the maximum speed. And this also makes sense because the more customers come in, the less free sandwich artists there are, right? Free sandwich makers there are. So if there's 4 customers and one substrate comes in, he has to go to the one maker that's available. He can't just go to any sandwich maker because not all of them are free. Only one of them is free. So as the substrate increases, you'll notice, look at this. The rate of change is very high here, right? How do we know rate? We know rate from the slope of the line, right? Oh, this is red. We get rate from the slope of a line, right? So this is much steeper than this, right? And that's because as the substrate increases, it has a harder time finding a free enzyme because there's less free enzymes. The amount of enzymes getting are, I think that makes sense. Okay, so now we get into like a little bit more mathematically. How does this actually make sense? So Vmax is the maximum speed that the reaction can proceed, right? The maximum amount of product that we can make, the maximum amount that the makers can work, the enzymes can work, right? Now we can represent this as an equation. The equation is Vmax is equal to the enzyme concentration times the K catalytic. Now this, if we convert it to our sandwich explanation, right? Our Vmax is the maximum number of sandwiches that we can make, right? Max number of sandwiches made, right? And what is our E? What is our enzyme in this analogy? Who is doing the work and making the sandwich? Sandwich maker, right? And then we have this new value, K catalytic, right? K catalytic means how much how much product does the sandwich maker make in one, one go, in one reaction, right? So in this case, we'd be like, how many sandwiches made for a customer, per customer? So we can think of it that way. So this, intimidating looking equation is basically just simple math. It's how many enzymes do I have? How much do they make if they do one go? Gives me the total amount of product that I'll get, right? And it makes sense. If a sandwich maker only makes one sandwich in one reaction, then if you have 5 enzymes and it's only one, then your Vmax would be 5. But if he's really good and he can make 2 at a time, and he has 5, then your Vmax would be 10, right? So it looks crazy, but I hope it's not actually that crazy. I hope that it actually makes sense in this way. So does that make sense? Oh, okay. Yeah, yes. Okay, good. Okay. So now what? Oh, actually, I'll leave that there. So now, the equation shows that Vmax is simply the maximum speed of all the enzymes combined, calculated by multiplying the concentration of enzyme by how fast each one works, right? Now, why does Vmax even appear? At a certain point, the amount of substrate is so great that the enzymes are already working as fast as they can. Once we reach this point of saturation, the addition of any new substrate will not make a difference in how fast the enzyme works, right? And that's basically what we talked about. If there's 10 customers, but there's only 5 enzymes, we're at the point of saturation. We can only make 5. And I guess this is another example, but with pencils and machines. I like sandwiches more. Finally, let's look at the Km on the Michaelis-Menten plot, and we talked about the substrate concentration at 1/2 Vmax, right? So, this is also really, really important. Km is the binding affinity of the substrate for the enzyme. So this is really, really, really important. A small Km indicates a high substrate affinity, and a large Km indicates a low substrate affinity. So let's look at the graph and kind of like convince ourselves why that makes sense. If the Km was really, really high up here, right? That means that the 1/2 Vmax is much further up. So let's say that the Km is way over here. And actually, no, that's bad. Let's say that the Km is over here. Okay, that's the worst straight line in the world. Right up here somewhere. Okay, and then let's say that our new 1/2 Vmax is over here, right? So this is my new Km, and this is my. Actually, I'm just going to draw an arrow. That's my new Km, and that's my new 1/2 Vmax, right? So if it takes a lot more substrate to get to the 1/2 Vmax point, so actually even forget about the 1/2, right? That might be confusing. Let's say it takes a lot more substrate, a lot more customers to reach maximum capacity. That means that the enzymes are working slowly, or rather that the enzymes are not binding to the substrate that well. They're not binding that rapidly. It's not that like a customer comes in and the maker immediately starts making a sandwich. It's more like the customer comes in and then the maker kind of ignores him for a while, and then maybe he starts working for him when he sees a bunch of other customers come in. So when the Km is high, it means that the binding affinity between the enzyme and the substrate is not that great. However, if the Km is really low, that means as soon as a customer comes in, the maker immediately makes a sandwich. It's like the good employees at Chipotle, right? Like, so as soon as they come in, they help the customer. right? Because they have a very good affinity between them. And that is going to lead to a lower Vmax and a lower well, not necessarily, but it's just going to be a very faster rate of reaction. So that is the fundamental explanation behind it. But it's very easy to, it's very important when we have those big kind of, this is a relationship that you need to remember, right? And there is an explanation that I just explained to you to remember it. However, the other thing that you should do is make a mnemonic that you can rely upon to remember, because you may get confused, right? So let's make a mnemonic to rely upon that allows us to remember this very well. So my mnemonic is, I remember Km. Km kind of sounds like the reason I remember Km is because of the game Mortal Kombat, right? So I think of Km as like combat or combativeness, right? So I think if there's a low amount of combat, then they're probably having high affinity. But if there's a high amount of combat, then they're probably not getting along with each other well. So obviously this isn't scientific at all, but when it comes to remembering, figure out the way that helps you remember these things, because you're not going to have the time or the brain power to like go back to the basics and be like, okay, let me figure out exactly why Km works the way it does, right? I'm giving you the explanation because it's important to know the explanation, but at the same time, memorizing this explanation in a way that makes sense to you, in a way that you will never forget it, is very, very important. Okay, any questions? Does that make sense? A small Km is high substrate affinity. A large Km is low substrate affinity for the enzyme. Does that make sense? Okay, good. Did I lose you guys with like the ridiculous sandwich analogy and the mnemonics? Catalytic efficiency. Okay, good. Awesome. Yeah, that's how I end up remembering those things in weird ways. So and then we have one more term, which is called catalytic efficiency, which is just a combination of the previous terms of the kcat that we originally brought up, which is the speed of our enzyme, the rate at which so turnover. What does turnover mean, right? How often are we turning our ingredients into a sandwich? That's kind of what turnover means here. So turnover number, kcat, divided by our km gives us catalytic efficiency. So let's think about this for a moment. This is actually very interesting, right? And this is just math. We can think of this as a ratio. We have our kcat, and then we have our km. So now, if let's say that our enzyme has really, really high affinity, right? If it has really high affinity for the substrate, the km is going to be high or low, right? We said it's going to be low. If it's a very small km in the denominator, then this overall thing is going to be large, right? Because it's an inverse relationship. If the denominator decreases, then the product increases. In this, oh, sorry, wait. Let me put it over the equation so it makes a little more sense. Or under the equation. So what I'm saying is, if I decrease my Km, that's going to increase my catalytic efficiency. And that makes sense because the catalytic efficiency is how effective the enzyme is at converting substrate to product. And if small Km, that means high affinity. So if it's a high affinity enzyme, it's going to be more catalytically efficient. That makes sense. Now let's think about kcat or catalytic, kcatalytic. So if kcatalytic, remember we said kcatalytic was the turnover number. How fast do we turn our ingredients into a sandwich? If we are very fast at turning our ingredients into a sandwich, right, we increase kcat, then we increase catalytic efficiency. Again, this makes sense because it's a direct relationship. Now, if we're very slow at turning our ingredients into a sandwich, then we decrease kcat, which would decrease our catalytic efficiency. So you see that there are two factors by which we can increase the catalytic efficiency of an enzyme. If we increase the rate at which we turn ingredients into a sandwich, and we decrease how fast we decrease the km, which means that we have high affinity for the customer, that's going to give us the maximum catalytic efficiency. So to give you I'll give you in the sandwich terms once, and then I'll give you in the scientific terms once. So we have a sandwich maker. The customer comes in. It's a low level of competitiveness between the customer and the sandwich maker. So they have a high affinity. This is all. So if it's a low level of competitiveness, which means that which means increased affinity. Whoops, which means increased affinity. Okay, so if it's a low level of combativeness, then it's an increased affinity between them. And then this sandwich maker is really good at his job. He's really good at flipping the ingredients into a sandwich. Therefore, he's very, very catalytic efficient, and he's very fast at getting the customer a sandwich, and he's out the door. And he gets a nice tip for his hard work or whatever, right? Now let's think about the opposite. Let's think about a really, not so good at his job. Let's say that this is a sandwich maker that's had a bad day at work, and he's very competitive. He's very passive aggressive to anybody that comes. He doesn't want to deal with anybody. Customer comes in, he doesn't even pay attention to the customer. There's a low affinity, right? There's a low affinity for the customer, and he's also not that good at his job. He's not really paying attention. He's not very good at flipping the ingredients into a sandwich. Therefore, we have a very inefficient worker. We don't have a good rate of we don't even have a good rate of taking the order from the customer, and then we don't have a good rate of making the sandwich itself. And that leads to very low catalytic efficiency. So that is the sandwich explanation. And now we're going to turn it into science, into enzymes, right? So we have an enzyme that has a high affinity for our substrate, So if it has a high affinity for our substrate, that means that the substrate binds the enzyme very, very quickly. That means that the substrate concentration at 1/2 Vmax is going to be low, because we reach 1/2 Vmax very quickly because we're binding them so quickly. So we have a low Vmax, and if we have a low Vmax, that translates to an increase in catalytic efficiency. We also have an enzyme that has a very good turnover rate. It takes the substrate and it flips it into product like very, very quickly. And so if that increases, that's also another way to increase catalytic efficiency. Okay. Any questions about the scientific version or anything? Is this making sense so far? Okay, good. And so then we have the opposite. If it's a slow enzyme, right? If it has low affinity, then it takes a lot of substrate to get to 1/2 Vmax. So it is a high Km. And if it's a high Km, that's inversely proportional, leading to decreased catalytic efficiency. And if it's a very slow enzyme, it's slow to turn over the product, then it's again a decrease. So that's again a decrease. Okay, so this scary equation is not really that big of a deal if you understand what each of these things means. I hope everybody was able to understand what each of these things mean, and it's in the Anki cards if you need to review. And then the last part of all of this. Oh no, this is not the last part of it. Actually, this goes for a while because we have to cover inhibition next. Okay, hold on one second. So Lineweaver-Burke plots are the same data, but they look differently because instead of doing the Vmax and the Km, we're doing the inverse. It's an inverse graph. So it's the 1 over Vmax and the negative 1 over Km. So this is a little bit interesting to get used to. Everything is the same. Again, this is still the velocity and this is still the substrate concentration, but it has been flipped into the inverse. So now when you look, the reason that this is really good, this is actually extremely good. Why is this extremely good? Because it gives us a very easy way to find this Km value and this Vmax value, because they are simply the intercept. The y-intercept is the 1 over Vmax, which, so if the y-intercept is 1 over Vmax, then if you flip it, you get the Vmax. And if the x-intercept is negative 1 over Km, then if you flip it and multiply it by negative 1, you get the Km. But it's a little tricky to keep track of because everything is flipped and negated and all this stuff. So you just have to keep your head on straight when you're dealing with one of these. And so when we talk, so then there's different types of inhibition of enzymes. So enzymes can become inhibited by let's see. Enzyme, yes, here we are. Okay. So we have here the other important part of the Lineweaver-Burke plot. So before we were looking at Michaelis-Menten plots with the curve, and then this linear one gives you 3 very important factors. X-axis, negative 1 over Km. Y-axis, 1 over Vmax. The slope is Km over Vmax. So those are the factors that you know, because they can give you graphs and ask you, like, you know, what's going on here. Then we have, oh, let's not start with mixed. Let's start with competitive inhibition. That's the easiest one. Okay, so competitive inhibition is when we have, so here's an enzyme, and this square is its substrate, right? So it binds the substrate and it makes the product, and that's good. If it binds this inhibitor, then it's no longer able to bind its substrate. And notice, where does the inhibitor bind? The inhibitor binds at the active site where the substrate would have bound, at this little square spot. That's what a competitive inhibitor is. It competes for the active site. The active site is the spot on the enzyme where it binds the substrate. So it directly competes for that spot. So what does that mean? A competitive inhibitor binds the enzyme only. If it binds the enzyme, it increases the Km. It increases the Km because when Km increases, affinity decreases, right? So if it increases the Km on the Lineweaver-Burke plot, since we increased, we go more to the right on the x-axis, because this is double flipped because it's inverse, but it's also negative. So it still ends up it still ends up going to the right, and so it goes to the right, and that is a competitive inhibitor. So if you see this situation, if you see a line here and a line here, they could just show you these 2 lines and be like, what kind of inhibitor is this? And you would have to know that this is a competitive inhibitor because the x-axis moved a little bit to the right. Now, that is the scientific explanation. The non-scientific explanation is that these 2 lines look like 2 swords crossing, and that's combat. That's a competitive inhibitor. That's competition. That's my mnemonic for recognizing a competitive inhibitor. So that is competitive inhibition. Then, so that's one type. That's the most simple type of inhibition. Then we have uncompetitive inhibition. So let's go back to the card. Uncompetitive inhibition is when the inhibitor is not going for the active site. It's going for another site on the enzyme, which is called the allosteric site. And now allosteric site just means it's a binding site that is not the active site. So that means that the substrate can still bind, but the inhibitor can come in and bind it as well. So this is going to this binds in a different way. So when an uncompetitive inhibitor binds, it only binds after the substrate has bound. So it's binding to ES only. When that happens, it decreases Km. This is a little weird. It decreases Km, which means it increases affinity. Why does it increase affinity? Because it's maintaining this enzyme substrate relationship for longer. It's preventing this from turning into the product. So technically, it's increasing affinity by decreasing the Km. And then the Vmax is going to decrease the same as the Km decrease. And I remember this as an uncompetitive inhibitor has a united effect on both of these. So whereas in a competitive inhibitor, there's no change on the Vmax. Why is there no change on the Vmax? Because if you simply put a bunch of substrate, you can outcompete the inhibitor. If you drop like 1,000, let's say if we go back to a sandwich, right? Let's say an inhibitor an inhibitor is like maybe like a delinquent that comes in and like messes around in the store or like tries to steal food and it like distracts like one of the sandwich makers, right? But if there's like, in this case, it's like if there's like 100 customers in the store, the delinquent's not going to be able to change anything, right? Like it's already, so there's too much substrate and the inhibitor can't really have an effect. So those are the main two types, competitive and uncompetitive. Then we have non-competitive. Oh, so wait, no, I have to talk about, for uncompetitive, I have to talk about what does it look like. So look, an uncompetitive looks like this because it has the exact same change on the Km and on the Vmax. So the way I remember uncompetitive is because between the lines, you can draw a U. So that's uncompetitive. That's an uncompetitive inhibitor. And then we have non-competitive inhibition. Non-competitive inhibition has some like specific details that you can dig into, but non-competitive inhibition is essentially that it can bind either way, I think. Wait, is that what it is? Non-competitive inhibition. Hold on. Mixed inhibition, the inhibitor binds to an allosteric site where the free enzyme, many of the binding is biased towards one or the other. Mixed inhibition can be quite complicated and is not often tested on the MCAT. True. This is a special case, though, in which 50% of the inhibitors bind the enzyme alone, 50% bind the enzyme substrate complex. This is non, okay, 50%, 50%, that's what it is. So non-competitive inhibition is like, it's not choosy. It doesn't have a preference. It can either bind the enzyme alone, or it can bind the enzyme after it's been bound to the substrate. And a non-competitive one looks like this because it didn't affect the Km, but it affected the Vmax. And again, the way the visual mnemonic for this is that you can draw an N right here, non-competitive. Okay. And so, oh, and then this is a nice little table summarizing everything. As we said, competitive, so competitive is the only one where Vmax is unchanged because the substrate can just outcompete the inhibitor. But the other ones, the substrate cannot just outcompete the inhibitor. The Vmax will decrease. This is the only one where the Km increases. Again, this works well with like the combat mnemonic because if it's competitive, then the combat is increasing. If it's uncompetitive, the combat is decreasing. If it's non-competitive, then the combat is about the same. And then the active site is the competitive, and the others are all binding to the allosteric site. Yeah. So this is a really big topic. This is a really, really heavily tested topic. Enzymes are super important. I hope that makes some sense. Any questions if it doesn't make any sense? Okay, good. then we're going to, I think this is almost at the end, regulating enzyme activity. This is temperature. Yeah. So temperature, there's an ideal temperature that you have to be at. If you're too cold, you'll die. If you're too hot, you'll also die. So you got to be at the ideal temperature for an enzyme to be perfectly in the perfect folding conformation to be doing its good stuff, right? If you increase the temperature a little bit, it's good because it makes the enzyme molecularly, things are moving around faster. But if it's too high, you're going to denature the enzyme. pH is the exact same thing. We're talking about, not denaturing it and not, you're going to denature it at either end. You need to be at the right optimal pH for the enzyme to fold properly. Same thing with pH. Covalent modification. Okay, so we hinted at this earlier. If you phosphorylate an enzyme, glycosylate an enzyme, meaning sugar it, if you put a methyl on it, if you put an acetyl group on it, it can change the function of the enzyme. Most common you see, like when enzymes are phosphorylated or dephosphorylated, it activates or inactivates them. There's not a one-to-one link. A phosphorylation is not always an activation. It's not always a deactivation. It depends on the enzyme. Allosteric sites, we talked about this already. Allosteric sites are, away from the active site, and they bind activators or inhibitors that, encourage the enzyme or discourage the enzyme. Zymogens are the inactive enzyme, and they need to be cleaved in order to be activated. So this is very important if we think about digestive enzymes such as trypsin or pepsinogen in the stomach, right? So if they're produced, when they're produced, they can't just start working right away. They're going to destroy the machinery around that was used to create them, right? Rather, they're created in an inactive form, then sent to the place where they need to be, and when they're there, they get cleaved, and then they start working and they do their function properly. Feedback regulation. Okay, so this is something else that we had talked about earlier, not today, but before. Regulation, so these are enzymes, right? Enzymes are creating products, right? So A is the reactant that gets turned into B gets turned into C. The product, if we have too much C, then we don't want to keep making more B, because B gets turned into C. So if we have too much C, then we don't want to turn any more A into B. So then C, the existence of C, tells enzyme 1, okay, let me reduce the amount of B that I make. This is called negative feedback. The product later in the line tells something earlier in the line, hey, you can slow down. That's negative feedback. Now feed forward, we have feedback, and now we have feed forward. Feed forward says, if the existence of A, let's say we have way too much A happening, right? If we have way too much A, then we need to convert some of the A into some of the downstream products. So A is going to activate A is going to activate enzyme 2 to convert some of the B into C. So when it converts some of the B into C, B is going to go down and C is going to go up. And then this is going to keep going until C is too high. And when C is too high, it's going to tell enzyme 1, okay, you can stop now. We are at a good spot. So this is how we maintain homeostasis. This is how, if we have an overload of A, we turn it into B, and then we turn it into C, and then if we have an overload of C, we stop from overloading C anymore, and then we even out. That's how enzymes and products regulate themselves. Cooperativity. Oh, I mean, okay, sure. Cooperativity is okay. So we talked about how some proteins have more than one subunit, like hemoglobin. Hemoglobin has 4 subunits, and each of them bind one molecule. The interesting thing is that when one hemoglobin subunit binds an oxygen, it encourages the next subunit to bind another oxygen, and so on and so forth. So that makes hemoglobin very, very good at picking up oxygen, which makes sense. If you pick up one oxygen, there's probably much more oxygen to pick up. So it makes hemoglobin very good at picking up the oxygen. And this is mathematically represented by this thing called Hill coefficient. Hill coefficient basically just means if it's greater than 1, then one subunit binding makes the next subunit more likely to bind. Negative less than 1 means if one subunit binds, then the next one is less likely to bind. That's the difference, right? So hemoglobin has a positive cooperativity between its subunits. If one subunit binds oxygen, then the other subunits are more likely to bind oxygen. And then equal, if it's just equal to 1, that means it's not cooperative. The binding of the substrate to the first subunit doesn't affect the binding of the second subunit. And this is a graph that shows you cooperativity, because as substrate increases and it binds, we see much faster binding. And this is called sigmoidal because it's an S. This basically just represents cooperativity in terms of a graph. And that's it. We've made it to the end. Any questions? Okay, okay.